Welcome to the GenePattern Notebook webinar. Over the next 40 minutes, we'll be introducing you to the features of the notebook environment and showing you how it enhances Jupyter Notebooks by incorporating hundreds of bioinformatics tools without the need for programming and extends the notebook interface to make it available to a wider range of scientists. We'll first describe the individual components of GenePattern Notebook and then show you how they work in an example single-cell analysis notebook. Then we'll demonstrate the GenePattern Notebook Workspace, where you can create, share, and publish your notebooks online. We'll describe some new features in development and then take any questions you may have. Our presenters today, in their order of appearance, are Chief Software Architect Ted Liefeld, Bioinformatics Scientist Edwin Juarez, Senior Software Engineer Thorin Tabor, and not presenting but behind the scenes monitoring your questions, Senior SQA Engineer Bobby Hill. About the presentation, if you have any questions, please type or paste them into the chat and we'll answer as many as we can at the end of the webinar. The software is developed in the lab of Jill Messerov at UCSD. In addition to GenePattern, the lab also developed Gene Set Enrichment Analysis, or GSEA, and IGV, the Integrative Genomics Viewer. These projects have been actively developed for over 15 years and have over 300,000 registered users. As many of you know, over the past several years, there has been an explosion of notebook environments. This started with the widespread adoption of Jupyter Notebook. Very quickly, derivative projects sprang up that aimed at extending its capabilities, such as Google Collaboratory, and connecting Jupyter with various cloud-based resources and repositories, such as Azure Notebooks, in addition to commercial and open-source organizations providing notebook hosting services. Why has the notebook become the de facto standard for data science? First, the code and the scientific narrative live in the same document, enabling a single research narrative that represents an in silico experiment. The notebook can serve as a platform for reproducibility, as a record of the exact analytical steps that were taken. The flexible nature of the notebook allows it to serve as a scratch pad while an analysis is going on, and for publication, the same notebook can be cleaned up and presented as the final analysis. Notebooks allow you to combine the tutorial aspect of an analysis with the code itself, effectively uniting the educational and operational aspects of software use. As electronic documents, they are easy to publish and disseminate online. One of the things that almost all of the environments I've showed you have in common is a limitation that prevents even wider adoption of the notebook environment. That is, in order to use notebooks, you need to be a programmer. You need to know Python, R, or some other supported language, and you also need familiarity with analysis packages and display packages. This involves a considerable investment of time. For those who want to analyze genomic data, there's another issue. There are a large number of software tools that are already available for bioinformatics. Some of these are command line, so you could install them into your Jupyter environment with all of their dependencies and maintain them that way. But that isn't making your life any easier, and it prevents your notebooks from being adopted by others who may be on a different platform. One additional concern is compute-intensive analyses such as assembly, alignment, or a number of highly parametric methods such as non-negative matrix factorization. You don't want compute-intensive analyses to be done in the Jupyter kernel if you can avoid it. To address all of these considerations, we've developed the GenePattern Notebook Environment. This environment gives you all of the notebook features of Jupyter and adds the ability to utilize analyses from any GenePattern server, which I'll describe shortly, in addition to a number of other general features to make it easier for, first, non-programmers to have access to the full range of genomic analyses required for today's integrative genomics experiments, and second, for the developers of notebooks to make it easier for their own notebooks to be more accessible to users at all levels of computational sophistication. The GenePattern Ecosystem is a platform for integrative genomics that supports tool interoperability and reproducible in silico analyses. It does this through a comprehensive set of analyses, including gene expression, sequence variation, and proteomics, as well as technology platform-specific modules with visualization tools for each available data type. There's the ability to add new tools without programming. Users can add tools to their own GenePattern installations to create their own specialized GenePattern servers. A pipeline environment records the analysis steps taken and can chain them into pipelines that can be shared, reproduced, and published, and interfaces are available for all levels of user. We released the GenePattern Notebook Environment in 2017, and we'll be discussing it in more detail. What kind of community has evolved from this ecosystem? GenePattern was first released in 2004 and has been updated continually. Our hosted server runs between 2,000 and 5,000 analyses per week. These run from simple variation filters to intensive RNA-seq analyses. There are over 75,000 users, both who use our hosted server and who have downloaded the software to use at their own organizations. Our community resource, GPark, has had contributions that have widened the scope of available analyses. Now we'll get started describing the features of GenePattern Notebook.
Now that you've learned a little bit about the GenePattern ecosystem, we're going to walk through a notebook that demonstrates many of the features of the GenePattern notebooks and what makes it different from a regular Jupyter installation. As we go through this notebook, the features I'm going to demonstrate will be called out in blue boxes like this one. To begin with, GenePattern notebooks add a what you see is what you get rich text editor. So normally, if you were to click into a markdown cell like this one, you'll see raw HTML as we have here, or raw markdown code. We have added the button here to open a rich text editor, which allows you to interact with the text as if you were in a normal word processing application or a Google application. So for example, I can adjust the formatting, and when I'm done, I simply hit run as with any regular markdown cell, and it will apply the text changes. Next, we have GenePattern single sign-on. So you already know that GenePattern has remote computation servers that allow you to run analyses that require more CPU or memory than is typically available on your laptop or on the GenePattern Notebook server. So this allows you to log into the GenePattern server, and the single sign-on allows you to log in directly just with one click as the same user that you've logged into this notebook as. Now I can also click cancel and select a different GenePattern server or use a different username if I choose. But the simplest thing is to simply use the same name, so I'll click login. And now I can run the remote analyses. Now, for the remainder of this notebook, I'm going to hide the scientific content. The next section will cover a scientific use case, but for here, we just want to focus on the GenePattern features. So I'm going to run this cell to just block out the text for the details. So, running remote analyses on the GenePattern server. When you wish to run a remote analysis, you'll get a form like this one, which allows you to set input data sets and the other parameters that are necessary to run that analysis. When you're happy with your entries, you simply hit the Run button, and that will submit that job to our remote server, which will then find a suitably large compute node to run it on, run the analysis, and when it's done, it will generate the results and bring them back to you in a cell like this one. Because this analysis takes about half an hour to run, we're going to use the tradition of the great baking shows and go to a different oven to pull out our already created results. Now, if you want to insert another analysis into your notebook, you do that from the tool catalog. That's available from over here, the thumb on the left-hand side, and also from up here in the Tools button on the toolbar. So when I click that, the catalog opens, and then I can go in and filter to find, for example, a analysis that I'm interested in, like Seurat preprocessing. Clicking that inserts a new cell with all of the various controls for adding my data sets and setting my parameters. Now, the next thing to look at is sending data from one analysis to another. So we have our DESeq2 results, and if we want to then send them to clustering, which we have already added to the notebook, we can do it a couple of ways. One is to click on the result that we're interested in, and then we say select send to existing GenePattern cell, and you'll see it comes up with the list of analyses that will accept that data type. I pick hierarchical clustering, and you'll see that it was automatically added into the correct input slot. I can also do that from within the hierarchical clustering or any module itself by clicking on an input type, and then it will look for matching analysis results that are already in the notebook and that I can select from, and that will add that directly to that parameter. Now, finally, let's talk about GenePattern visualizers. So these are actually interactive JavaScript visualizations, and you run them and insert them into your notebook the exact same way as you do the remote analyses. But when you hit run, things are a little different. Instead of submitting a remote analysis to our servers, it's going to bring the visualizer in this new frame for you so that you can then work with your data in a more interactive form. Since the Jupyter environment puts some constraints on us for the amount of space available, we've also added another feature that we call pop out. So from the tools menu, I can come up here and say pop out visualizer, and that will open up that visualizer for me in a new fresh window where I can use the entire screen to interact with my data. Next, let's look at a few of the programmatic features that are available. So while all of the analyses that we've shown you till now have been done on the remote server, sometimes you might want to load it into the Python kernel as well to work with it directly. So when you have an output file, like the ones we've seen before, you can click on it. And instead of sending to another GenePattern cell, we have a few other options. Open a new tab. We'll just open that file in a browser so you can look at it. Send to code. We'll insert a new cell like this that gives you essentially a Python file-like object that you can use to then download and work with the data. But more useful, for many data types, there is also a send to data frame menu item available. This will actually create another cell that downloads the data and actually inserts it into a pandas data frame object that you can then perform analyses on or look at, as we see here. Now, throughout the notebook so far, you've been seeing the user interfaces that are available in the notebook. These are created using the UI builder. So, for example, this cell here, you'll see that what we've done is we've put this little annotation at GenePattern build UI ahead of a Python function. Now, when this cell is run, what it does is it will generate this user interface for us so that when I then want to use it, instead of executing the code cell, I can hit the run button and it will run that function for me. In addition, when it has created a UI builder cell like this, it will have added it to the tool catalog. So for example, if I go up and click on Notebook, you'll see my Hello World function here, and I can add another copy of that as many times as I want, same as adding a analysis from the remote server. Now this works also for working with R code as well. The syntax is slightly different. You see here we use a cell magic instead of a Python annotation. And in this case, we have a little R function that's simply going to plot some random numbers. And when we run that, you'll see that it runs the R code, creates the plot, and presents the results in the notebook. Finally, there are other tools that can be connected into GenePattern notebooks as well. One of particular note is the Integrative Genomics Viewer, which is also from the Mezarov lab here at UCSD. So for example, here I have a little UI builder cell, and all we've done is set it up so that you can open up a particular GATK dataset on a particular gene. So when I hit run, you'll see that it creates an IGV browser and then goes out, fetches the remote data, and loads it into that IGV browser. This concludes this section. In the next section, you will see a scientific use case that uses many of these features.
So now that you're familiar with some of the basic elements of GenePattern Notebook, we're going to use those features to perform some scientific analysis. Particularly, we're going to be analyzing single-cell RNA sequencing data utilizing Seurat, which is a very popular tool written in R. And we're going to particularly be highlighting how easy it is to perform this analysis using Seurat in GenePattern, whether you are comfortable programming or not. And in order to make things a bit easier to follow along, we have separated the Seurat pipeline into their building blocks that we're calling modules. Now, in this notebook, we're going to be using the 10x genomics file format as our primary input. We could have used other input files, but that one's common enough that that's the one we're using to feed into the first of the modules, which is Seurat QC. If we had multiple inputs, we could have used Seurat batch correction. Either of those modules produces an output that is compatible with Seurat preprocessing, and that module itself creates an output that we are going to feed into Seurat clustering. In the future, we're going to release a module called Seurat attack sequencing that will allow us to add attack sequencing data to our analysis. So the first thing that you want to do, as always, is to log in with your username, and you will have that already pre-filled for you. So now that we are here, let's talk about what the actual data that we're gonna look into is. We're gonna be using these peripheral blood mononuclear cells, or PBMCs, that 10x Genomics makes available. You may have seen it in many other analyses. This is what the Seurat vignette uses, for example, and these are 2700 cells sequenced by Illumina NextSeq 500 and run through the Cell Ranger pipeline. So something very similar to what you may encounter in your day-to-day analysis. Particularly, we're going to be using the same output files that Cell Ranger uses. And we're providing those in a hyperlink over here. But if you have a zip file with those files, that is precisely what we're gonna be using in here. And just a note on what those three files are, if you've never seen them, they always come in these three, at least for the outputs of Cell Ranger. One is a matrix.txt that has the gene ID index, cell barcode index, and the total counts. And there's a barcode file, a TSV file, that references those indexes and gives us the barcodes of each individual cell, and a features.tsv file which has the names of the genes that were referenced by the index in the matrix.mtx. Sometimes this is called genes.tsv, but generally it's features.tsv. And as we had discussed previously, the steps that we're going to be follow are the three steps that we will normally follow in any analysis. So it will be, we're going to start by doing some loading the data and visualizing some QC metrics. We're going to take some notes on some of those QC metrics, using that for data filtering, normalization, and dimensionality reduction in order to then do clustering. So that is more or less going to be the workflow that we're going to be following in here. And so let's go ahead and get started. The first step will be to take a look at the QC metrics. Now, why do we want to do that? That is because we want to make sure that our data it's looking decently, that there are no glaring problems, and if there are some problems, then we can filter those out in the next step. So we're going to be using three very commonly used markers for quality, which will be giving us the total number of genes that were detected in each cell, the total number of reads in each cell, and the percentage of those reads that map to the mitochondrial genome. We're gonna talk a little bit more about how to use those in a second, but I wanted to give you a preview as to what these look like. These will be in the form of violin plots, so we can spot any potential outliers from the distributions. And of course, you could be doing this in R. If you're familiar with it and if you're comfortable programming, you could write a code like this one. This is the one that I was using to test this data, and it's actually the underlying code, the very same code that is going to be run in the next step. But instead of writing R, you can just run your module on GenePattern, which is going to be using the exact same code, but it's going to be running on the Amazon Web Services. So you're not going to need to worry about the performance of your own computer. So let's take a look at the inputs that you're going to need to provide. And because this is the QC module, we don't need a whole lot of inputs. The most important thing will be obviously our data, and you can leave the rest of the parameters by default, unless you know that your data was formatted in a different way. This is programmed to read in the 10x Genomics data the way that it is. Again, if you have a different type of data, then feel free to change the rest of the parameters. And one way to provide your data is to either upload a file from here. If you have compatible files loaded into the notebook, they will be show up over here. We're gonna see that in just a second. But another way would be to add a URL, and an easy way to add an URL will be to scroll up and then drag and drop from here. So you can grab our file or hyperlink, drop it in here, and then we will be able to run the module. Again, because this is very straightforward, it's a QC module. The most important thing about it are the outputs. So we're going to go ahead and just run it. So you can hit run and then wait a few more minutes for this to be done. Because this is a video, I can use the magic of editing, and then I'm gonna wait a little bit for this to be done. So once the module is done running, we're going to be presented with this cell that has all the outputs from the module. And the most important output from Seurat QC are the plots that it generates. These are the QC plots. So we're going to take a look at it inside of the notebook. So we're provided this function that displays a PDF. And like I mentioned earlier, if you click on this area, you're going to see whether or not there are files that are compatible with this function. And in this case, we have this rplots PDF file in here. So if we click on it and then run this, we're going to see the outputs of the module. And these, like I said, are some of the basic QC plots that we're going to take a look at. And from here, we're going to decide what are going to be the next steps. So do we want to filter cells based on how many outliers do we see over here? Or are we going to filter cells based on the percent of the cells that are mapped to the mitochondrial DNA? Or maybe do we have like two cells that are showing too few genes that are being expressed? So those are the three metrics that we're gonna take a look at. And then here we can decide, let's say, let's just cut off cells that express more than 2,500 genes and maybe less than 200. So we wouldn't see that many outliers in here. For the mitochondrial ones, I estimate that 7.5 threshold just to get rid of some of these outliers. And we're going to do that in the next step. And as we can see here, I'm just summarizing the same conclusions that we drew. So the next step is going to be filtering and normalization based on the parameters that we just drew from the previous step. But let's take a second and discuss as to why we're going to take each of these steps. So the first one, it's kind of obvious, and we've been talking about this. We want to make sure that we filter cells that do not meet our quality control criteria. So you always want to make sure that you're doing this, but there are also other steps that are important to do in preprocessing, such as normalizing and scaling your data. Depending on the algorithms that you're running, they will make assumptions as to has your data being log normalized, or is this a Gaussian distribution? Is the scale between 0 and 1? Are they is the mean 0? So those kinds of preprocessing, we need to make sure that we're doing them. And in this notebook, all of the preprocessing steps are being taken, knowing what the downstream analyses are going to be in the next module. So we're doing the appropriate normalization and rescaling. If you want to look at the details as to what those are, you can take a look at the documentation that we provide both in the notebook and in the module. But we're going to be doing log normalization and doing a scaling of the data so that the mean is 0 and then the variance across cells is 1. So we're doing some rescaling of the data because that is what our PCA implementation, or the one that Seurat has, assumes. And then if you're not familiar with PCA, this is a method for dimensionality reduction. It stands for principal component analysis. I'm sure all of you have heard about it. It's very popular because it's really good at finding what are the dimensions within our data that are contributing the most to the variance in the data. Because single cell analysis, single cells data are super heterogeneous, this step becomes very useful, and we always want to do PCA before we go into the clustering step, which is going to be the next one. So that is why we're doing these steps, and that's all taken care of in Seurat preprocessing. So you don't have to worry about, oh, did I normalize my data, or should I have like log normalize it, or is PCA expecting it to be this way or this other way? We've already worried about that and implemented it this way. So similar as before, we want to use the input from the previous module. So we just click on here and we see this the output from the previous module in here. We're going to then make sure that these parameters meet our criteria. In this case, they happen to be what we wanted, because the defaults for this module match the sample data. But for your own data, you want to make sure that you're doing the filtering that you want. Either you may want to be more stringent about it, or less. And if you want to change some other parameters that are a bit more advanced, if you know the kind of normalization you want, and you're going to be doing some other kinds of analysis, you can also do that. But by default, you may not want to do that. And the same as before, you can then hit run and maybe wait 5 minutes or so, and your module is going to be done running, and then we're gonna see the outputs later. So again, we're just gonna wait 5 minutes here or so until this is done. So that took about 5 minutes in real time. Now let's take a look at the outputs of Seurat preprocessing. Similarly as before, we have a set of figures that we may want to take a look at, and also a file that we can pass to the next module. So let's use the same function that we used before to display. In this time, we're gonna use the second PDF, because this is the second module in the same notebook. And we're gonna hit run, and we're going to take a look at some of the outputs that Seurat preprocess creates. The first one is a volcano plot that shows us some of the most variable genes. So we can already start to take a look at whether some of these are either maybe what we were expecting or not, depending on the type of analysis that we're doing, right? Is this exploratory? Are we trying to corroborate a hypothesis that we have? So this kind of volcano plot can also be useful in your paper if you would like to use it. We have a PCA plot that shows us the top two principal components, just to see if we can start to already see some kind of clustering that may be happening in the next steps, right? So again, this is pre-processing, so a lot of this data is preliminary, so to speak. And again, this is some of the variability that we may see in each of the principal components associated with different genes. So there's a lot of information that we can take a look at. This is one of the most informative plots, in my opinion. It shows us the principal components. This is a parameter that we can change in the module, and it shows us the amount of variability that they contribute. So in the future, in the next step, we're going to take a look at this and decide how many principal components we're going to use in the clustering step. So in here, in between the 15 to 20 range, we're seeing diminishing returns, that the variability that they provide is not as large. So we don't need to use 50 or 100 principal components. For a different data set, maybe that's gonna be useful, or maybe for another data set, we will see that only using fewer components is the way to go. And finally, these heat maps show us very similar results as before. They give us this visual representation of the variability that each component shows. So particularly at around principal component 15 or so, this starts to look a little bit random to me, especially when compared to the first few principal components that show a lot more variability. And that's the kind of information that we want to take a look at. And then we're going to use those outputs and the information that we gain from that for the next step, which is going to be to cluster the cells. And because Seurat uses a graph-based method, particularly they use KNN and the Louvain method, feel free to read more about those. But the important thing about those graph-based methods is that the number of dimensions that you use to cluster them is very important, which is why we took some time to take a look at the PCA results, because that's going to be one of the parameters that we're going to use in the next module. But let's also take a look at another GenePattern feature in here, which is to send the outputs from one module to the next, rather than taking them from the previous one, right? So in this output cell, you can click on it and then send to existing GenePattern cell, and then we can send this to Seurat clustering. And then now we are populated the input Seurat RDS file. And we can change the rest of the parameters, and in particular we want to focus on this maximum dimension, which is the one that we selected from the PCA. This is the number of dimensions that we're going to use to cluster, or the maximum number of dimensions. The UMAP is the method that we're going to use to project our clusters, and this resolution is related to the UMAP method. Same with the number of markers. This is going to be actually the one of the outputs of the module. How many markers per cluster do we want to output? So let's go ahead and click run, and then we can wait for this to be done. So Seurat clustering is the module that takes the longest to run in the Seurat suite, and that would make sense since it's the one that it's doing the clustering for all the cells given the input that we've given it. And this one took about 10 to 15 minutes for the sample data. So if you provided larger data, it may take a little bit longer, but it's also not an outrageous amount of time to wait. And we can treat the outputs of this the same way that we treat the outputs of any other module. So in here we can view the plots that it creates in a PDF file with the same function that we have been using before, that shows us some of the clustering that this is doing. And we can already grab this and show it either to our colleagues, or put it in a paper as like the results from clustering. But you may want to do further analysis with this. In fact, I'm curious to see what the markers are for some of these clusters, and can we do that? And the answer is, of course, yes, we can do that. And for that, we are providing a function that you can either modify yourself, or you can write an equivalent function if you want to do something slightly different. And the first step will be to grab one of the output files from the GenePattern server that was created by the previous module, and put it in your workspace. So that's all that this function is doing. But if you don't want to look at the code, you don't have to. You can always toggle the code view. I'm doing this and exposing some of the code in here to show you that whether you are very experienced in programming and you want to develop a notebook for someone else that's looking clean and doesn't have the code exposed, you can do that very easily here in GenePattern. Or if you are on the user end of the notebooks, you don't have to worry about the code, but you know that it's there, and if you share it with a colleague, they will be running the same code that you were running. So in here, let's grab the output from the last module and run this function that I just described. So because it's accessing the server and grabbing this file that it's relatively large, it may take a minute or so to run, and it lets us know when it's finished. So we can go ahead and start using in this file here and read it into the next function, which it's a very simple R function, and notice that we're using both Python and R on the same notebook. And I think this is one of the most powerful and useful features of GenePattern, that you can choose the language that you want to use, or you can use both languages that are the most popular ones these days, or you can use neither and stay with the graphical user interface. I personally use all three in the notebooks that whenever I'm doing my own analysis, as you can see over here. And I'm showing you the code so that you can see that, right? But we can always easily toggle the code view. You can do that with all the GenePattern cells. So we're going to be using this function to annotate some clusters, actually to visualize those markers. And we can take a look at these two markers. And why did I select those two markers? Well, for two simple reasons. First, I knew that these two markers were going to be highly expressed in only one of the clusters. And second, we know that they are markers for these canonical cells that we can find in this dataset. MS4A1 is a marker for B cells, and CD8A is a marker for CD8 T cells. So this is what we would have expected, right? That the marker for B cells is overexpressed in this cluster, in cluster 3 in this case, and not very expressed in the other ones, except for maybe some outliers. The same for CD8A, and we can look at not only the violin plots, but also these cluster plots that is the same clustering that we have shown before, but now we're annotating where those cells are being expressed, and we can overlay these with the clusters that were shown in the module, put that in our paper, and show that these markers in clusters correspond to the same one, annotate them, and move on from that. And finally, we are providing a function that remove the intermediate files for this notebook. You don't have to run this in every notebook. I like running it in here. And I'm also showing you the one last feature that I wanted to highlight, which is if you can just run straight up code in GenePattern, as we have seen before. If you encounter that and your intention is to run the cell, you can always run that. This is a cell that was designed to then show the graphical user interface. This is for those of you who may feel more comfortable programming and want to develop notebooks for other people. If you're going to be running notebooks that were created by the GenePattern team, you will rarely see code exposed like that, because our intention is to use graphical user interfaces in general. But you can just click run, and this will remove these temporary files that may just take up space in your workspace, and you may not want them laying around. So that concludes this preliminary analysis on some single scRNASeq data, and feel free to use this notebook for your own scientific endeavors, and I personally will be happy to answer any questions that you have, either about any of the features that we didn't highlight here, or any features that maybe we haven't implemented yet, or some questions as to how can you use GenePattern to run analysis on your own data. So thank you.
In this section, I'm going to be talking about the GenePattern Notebook Workspace. Put simply, the GenePattern Notebook Workspace is our free and public resource for running, authoring, and sharing GenePattern notebooks. You can access it right now at https://notebook.genepattern.org. It's an easy way to try out all of the GenePattern Notebook features you've heard about with no installation required. And did I mention it's free and open to the public? All you need to do is sign up for a GenePattern account. When you log in, you will have access to our extensive notebook library. This library includes notebooks for many common and popular bioinformatics methods, including methods for working with RNA-seq and single-cell data. It also contains a variety of tutorials, workshop materials, and templates for different workflows. All of the notebooks in the library can be previewed without the need to run or download the notebook, or even log in. This can be accomplished from each notebook's preview page, which includes the notebook's name, description, author, and a full screenshot of the entire notebook. You can also use this page to run or download the notebook, respectively. In addition to the notebooks that are already in the library, you can also easily publish or share your own notebooks with other GenePattern users. Just select the notebook you wish to share, and then click the Publish or Share buttons. Publish lets you attach a description, and then adds your notebook to the existing library. Share allows you to selectively share your notebook with specific co-authors or collaborators. These provide an easy way to develop notebooks with others, to publish a notebook to accompany a paper, or to otherwise enable other users to utilize your method. In addition to the notebook library, the GenePattern Notebook Workspace comes with a curated set of programmatic libraries that are already pre-installed. These include all of the Anaconda data science packages such as Pandas, Plotly, Matplotlib, NumPy, SciPy, and scikit-learn. It also includes IGV, Bioconda, Bioconductor, Seurat, and many more. Furthermore, if you need a library that's not pre-installed, the GenePattern Notebook Workspace allows you to install your own libraries as well. Just invoke pip, conda, cran, or bioconductor and install what you need. This can be done from the command line or even directly from a notebook. In addition to the curated collection of notebooks and libraries, the GenePattern Notebook Workspace also comes with a curated set of extensions that make working with notebooks easier. This includes the Table of Contents extension, which you may have noticed in Ted and Edwin's earlier presentations. This extension dynamically generates a table of contents based on the notebook's headers, which is then displayed on the left. You can even click on an entry in the table to skip to that particular section in the notebook. Another extension that comes with the GenePattern Notebook Workspace is collapsible headers. This provides the ability to expand or collapse different parts of a notebook based on the notebook's headers. In the screenshot, you can see that these Step 2 and Step 3 headers are collapsed, while Step 4 and Step 4a are expanded. Collapsing a header hides all of the cells between it and the next section. This is denoted with the ... indicator you see on the right. Expanding a header reveals those cells. And of course, the GenePattern Notebook Workspace comes with all of the GenePattern extensions that Ted went over earlier, including the Rich Text Editor, UI Builder, and GenePattern Cells. And just as a reminder, all of this is free, open source, and publicly available at https://notebook.genepattern.org. Just point your browser there and log in. Now, some of you might be working with protected medical data, and you may not feel comfortable running your analyses on a public workspace. For those of you where that's the case, or if you just prefer to run things in your own machine, you have the option to download and install your own GenePattern Notebook instance. The full stack is already set up and installed in a Docker container, and that's available through Docker Hub. Just pull GenePattern/GenePattern-Notebook. Or if you're a more technically savvy hands-on sort, you can install each of the individual packages using either the pip or conda package managers. If you install the GenePattern-Notebook meta package, you'll get the entire suite. We're always improving our GenePattern Notebook offerings, and in this next section, I want to talk about some of the upcoming features that we have in development. You may have heard of Jupyter Lab. It's the latest and greatest interface for Jupyter, and it comes with a variety of improvements and new features, such as improved customizability, themes, layout options, and other user experience tweaks. GenePattern Notebook's support for Jupyter Lab is well underway, and it will be released in the near future. We've also been experimenting with what we call our Notebook Project functionality, which is the ability to encapsulate individual notebooks and their dependencies in a portable environment, so that when you launch a notebook, it comes with its own libraries supporting files and other necessary elements. This helps improve reproducibility and provides an easy way to support different environments and tools. It also helps improve the GenePattern Notebook Workspace's publishing and sharing capabilities. Notebook projects will be released in the not-too-distant future. Finally, we've been working with the Galaxy and Cytoscape teams to integrate these two bioinformatics tools into the GenePattern Notebook ecosystem. Once completed, you will be able to run Galaxy jobs or view Cytoscape networks in much the same way that you can currently run GenePattern in the Notebook environment. So before I finish, let me just give a few acknowledgements to people who have contributed to GenePattern and GenePattern Notebook. You can see the team on the left. Jill Mezarov is our PI. Funding has been made possible through the National Cancer Institute's Informatics Technology for Cancer Research. I would also like to put up a few resources from the net which you can visit if you want more information. These include the GenePattern Notebook Workspace, our website, our GitHub organization, Docker Hub, and more. Finally, I would like to thank everyone for listening to our webinar.